My name is Ari Helenius, and I'm from the Institute of Biochemistry at ETH Zurich in Switzerland. In the last part of these seminars, which I have entitled Open Sesame, Cell Entry of Vaccinia virus, the focus will be now on a single virus and a virus and its entry into cells. It illustrates, what I will talk about, illustrates many of the things I've talked about before: binding, signaling, endocytosis, and so on, but in a very dramatic way. This virus is rather unusual because it's a very big virus. It's the most complicated virus family known for animal cells because it belongs to the poxviruses. Poxviruses are these giant viruses right here. They are enveloped animal viruses, and many of them cause diseases in different species. Perhaps the best known is smallpox, which is has been a major pathogen in humans. In fact, between 1914 and 1978, when this virus was finally eradicated, it's estimated that some 300 million people died of this virus. But as I mentioned, it is now eradicated due to a worldwide, smallpox is eradicated due to a worldwide vaccination program. And in fact, it's the only human virus that has been completely eliminated from the population. Now, the particle I will talk about is the so-called mature virion particle, because this virus, in its complexity, comes in many forms, or several different forms. The mature virus, of which you see an electron micrograph in the back here, is of Vaccinia virus. This is the vaccine strain of virus used to eradicate smallpox, and it is the most abundant form of the infectious virus that exists after infection. What you should know is that it is an enveloped animal virus. It has, unlike the other forms of the virus, only one single membrane. You can see that this light band here, it is a single lipid envelope. It's a DNA virus. It's very big, as you saw, compared to many other viruses. And it replicates in the cytoplasm, even though this is unusual for a DNA virus. Receptors for this virus have not been characterized in great detail yet, but it's already known that this, like many other viruses, uses heparan sulfate proteoglycans as an attachment factor. It is endocytosed, like many other viruses, in large vesicles. One can see those by electron microscopy, and it's known to be acid activated. So it has a membrane fusion protein complex which needs acid to work. So it looks like a normal virus, only it's much bigger and more complicated. So just to go through the early stages which we are interested in here, the virus binds to the cell surface like other viruses, is then internalized in large vacuoles. The acid inside these vacuoles, after they have formed, then induces membrane fusion. And the capsid, which is this dumbbell-shaped structure in the middle, is released into the cytosol, where replication or transcription of the first viral protein, a messenger RNA, starts inside the capsid. Later on, further ones occur from the naked DNA. So we are interested in how this virus enters cells. The work started when Jason Mercer, a postdoctoral fellow from the United States, arrived in the lab, and he had a viral particle which was labeled such that it's fluorescent. One of the core proteins, capsid proteins called A5, had a GFP, green fluorescent protein tag. And the virus is perfectly infectious but fluorescent. So the first thing he saw was that this virus also surfs on filopodia. So the host cell is out here, and you can see the virus moving along filopodia. And for all we know, this is also happening through actin retrograde flow, like the papilloma virus. And this phenomenon, as I said, was first seen by Walter Mottes and co-workers for other viruses. Here, however, something else happens. Follow what happens when the virus over here arrives at the cell surface. So this is speeded up. So after virus arrival at the cell surface, a bleb forms from the plasma membrane of the host cell. It grows for about 20 to 30 seconds, and then in the following 20 to 30 seconds, it retracts. And the bleb forms first where the virus is located, and later on all along the cell surface. You can see that spreading. Here again, the same virus is down here, and you can see the blebs moving up the cell body. So the whole cell becomes globally modified. That plasma membrane starts to blow out in blebs in many different places, up to 120 blebs per cell. So obviously the virus here is inducing a change in the bleb. It's probably inducing a signaling pathway, activating something which induces the formation of membrane blebs. Such blebs have been seen before in other situations which do not involve viruses, but not for viruses. And what we know about this blebbing is that it's very similar to what one finds during cell division, apoptosis, cell motility. A new technique called FibSem, of electron microscopy, has allowed our co-workers at ETH to look at this phenomenon in 3 dimensions using electron microscopy. And what you see here is going through a thick section of cells with the viral particles shown in quite light color. It's better appreciated if we look at it in 3 dimensions. The viruses have been pseudo-colored red here. And what you see is that the cell surface is modified. You have these blebs forming, and the viral particles seem to be endocytosing on the back side of the blebs into the cell. What you also can see here are the filopodia, which extend out, along which the viruses move towards the cell. So this is very unusual, but it enforces the idea that this virus is triggering a change in its host cell, complex change. That change involves actin very clearly, because the actin itself, in this case RFP label, moves into the blebs, and so does many actin modifying and actin regulating proteins. That is already known from bleb formation in other systems. All we can conclude at the moment is that the virus induces something that the cell also does under normal conditions. At the same time as Jason was looking at this plasma membrane phenomenology, he was collaborating with a group of Lucas Pelkmans in our institute at ETH in trying to identify the Vaccinia virus infectome, as we call it, basically to obtain a full list of cellular genes and cellular proteins involved in assisting the virus through its replication cycle, particularly in this case through the early stages of the replication cycle. So we wanted to have a full list of these Trojan people here who were helping the virus in. We want to know exactly who they are, what they're doing, and what their addresses are, and so on. Find out in what way and the molecular assistance given to the incoming virus happens. And this, to get such information today, is possible due to 2 completely spectacular events that have happened in the last 10 years. First of all, we have now access to the human genome sequence. Basically, we have the names of the Trojans. And we have a technology which allows us to eliminate 1 gene at a time and then test the cells whether they can be infected or not. And that is using high throughput siRNA silencing in human cells. A screen in which we silence genes 1 by 1 and test for infectivity. So this work has to be done in an automated setting, and it was done together with Lucas Pelkmans, Bernd Schneider, and Raphael Sacher at ETH. So basically you need a robot to deal with the technology, also the infection, all the work is done automatically. And then we need a library of siRNAs. In this case we used a library of 7,000 druggable genes. They are selected as being more amenable to future drug development than the others. So it's not the whole genome. In 384 well plates, the infection was tested, and we used 3 siRNAs per gene in triplicate. Moreover, we expressed in this case mature virus, Vaccinia, in which the GFP was included so that it was expressed from an early intermediate promoter. So when the cell got infected, it became green. And then readout was using an automated fluorescence microscopy setup where pictures were taken from all the wells, and then you will see in a moment how that can be used to detect infection. So we defined hits as those which had 2 or 3 out of the 3 siRNAs cause either 50% or less infection in cells compared to controls. This is how it looks. Here is a cell culture. Perhaps you can see the blue Hoek-stained cell nuclei, and the green ones are the ones which have been infected. This is in the control. With a control siRNA, about 10 to 15% of the cells are infected. Now, in a few cases, when we looked at them, we found that there were more cells infected than in the control, but this was not so common. In many cases, the good hits, the strong hits, looks like this. You can still see a few infected cells, but most of the cells are no longer infected in cells transfected with this particular siRNA. In this particular case, we have silenced the kinase called PAC1. I'll come back to that in a moment. So in the hits, we had 142 hits out of 7,000 with 3 out of 3 siRNAs, and 284 with more than 2 or 3 out of siRNAs, and then only 4 where the infection was actually increased. Now, what you get from this is then a big list of gene names, and there are many ways of looking at this. One is to make tables and pie charts like this, and you can see that when we identify which families of proteins these hits fall into, there is practically every family that cells have, from transcription factors to membrane transport factors, ion channels, proteasome components, etc. So this is not particularly helpful, except it tells us that the background to this infection is complex. A little more informative is if you combine the hit list with information known about interactions between proteins. And that can be done by using the string EMBL program, which allows you to take every hit, which every little ball here is a hit, and then see is that particular protein known to interact with anything else. And now you can see already some clusters forming. This cluster here has practically every subunit of the proteasome complex. This one has many subunits of the ribosome and then associated proteins and so on. So now we're starting to see some contours of the biology, the intracellular biology of this virus. For every virus we've done this, it looks different, and we're coming back in a moment to this cluster that has to do with tyrosine kinases and molecules involved in regulating actin dynamics. But before that, I'd like to pause a bit and think about this approach, because we and others are taking it, and I think it's going to mean a huge difference in the infectious disease area. So we are now looking at pathogen-host interactions using a systems biology approach. And by doing these screens and looking at which cellular components are important in supporting the infection of a given pathogen, we arrive at a huge information set, which can, if we can analyze it correctly, reveal critical host factors and processes that support or inhibit the infectious cycle of a pathogen. We know already they look different for different pathogens, but they are sometimes related. This information also exposes new molecular mechanisms in the replication cycle. We had never had a clue that the proteasome was involved. As you saw there, it's one of the major hits of the screen. The data also provides a basis for a functional classification of pathogens. We can now classify on the basis of these hit lists viruses according to their functions in the cell. different viruses. And it is also starting to give us some information about the cell and species tropism. Why are some cells infected, others not? In addition, I think that whole data will start to show us new similarities between viruses and viral diseases. And hopefully we will find common cellular factors involved, which may allow us to approach antiviral agents so that they can work on more than one virus at a time. So, of course, it provides us new approaches to perturb infection and obviously new potential targets for antiviral strategies. So I think this systems biology approach will be a key to a new stage in infection disease. However, where we now are, after we have the screen, we have to go back and do the cell biology and the molecular biology. We have to validate the different hits and clusters. So what we decided to do was to focus on one of the clusters, which is we call the PAC1, RAC1 clusters. These are various hits. PAC1 is a kinase known to be involved in regulating the dynamics of actin in cells, and RAC1 is a GTPase that activates PAC1. These other hits all make sense, and they are regulators of these two components. Now, the first thing to show, of course, is, and I won't go through more than very superficially, you have to use other methods to show that PAC1, for example, is a real hit. You have to use additional siRNAs. You have to use specific inhibitors, which we luckily have obtained. You can then use DNA dominant negative constructs to look at it. All this has been done. You can demonstrate that when you add the virus, this PAC1 actually gets activated, and so on. The main point here is that you have to move back at this stage to the cell biology and the biochemistry of the system, and then in a tedious set of experiments analyze it from that point of view. Here is just an example of what happens when you add the virus. Then the PAC1 GFP, which is shown in green, moves. This is a non-infected cell that's mainly cytosolic. Now it moves into the blebs and is there as long as the bleb exists. So PAC1 is indeed activated here. RAC1 is also activated. One can follow its activation. Already 5 minutes after addition of the virus, RAC1 is activated very heavily for about half an hour. Now, you can then also look at what the blebbing means. Is it essential for infection or not? And you can see if you use a dominant negative RAC1 construct, you block blebbing. You also block infection. It's not shown here. If you have a constitutively active RAC1, you increase blebbing and also infection. Another approach is to use chemicals. I mentioned this is in an earlier lecture. That's very powerful today. Inhibitors have their problems, but taken as one of the many approaches, they are powerful. Here we're looking at inhibitors of actin dynamics and inhibitors of different kinases here. And you can then get a picture of which components in the cell are important, which are not. All this can then be fitted into the known signaling pathways, and without going through any of the details here, I'll just show you here a cartoon of the various signaling pathways known, or the components known to modify, to regulate actin modifications down here by interactions happening at the cell surface. So that can go through 3 classes of receptors, receptor tyrosine kinases here, integrins, and trimeric G protein receptors here, which feed into this complex network of signaling factors. I'll just show you here which of these we already know from our work and other people's work are involved in the activation of the blebbing and the infection by Vaccinia mature particles. So it looks like it's coming probably from the receptor tyrosine kinase pathway towards the actin. This is obviously a cellular signaling pathway. The viruses are simply using it to trigger this cascade of events that eventually leads to their uptake. There are bacteria that also manipulate this same pathway, but they very often inject effector molecules into the cell, which then directly interact with these pathways and modify them so that they can be helpful for to the bacteria. Viruses have nothing to inject. They have to simply take advantage of the existing machinery. And this, I think, is an example of that. So what is the pathway like? We have a rudimentary picture. The receptors are not clear. Perhaps they involve to some extent EGF receptor because it gets activated. Then there are serine threonine kinases, PI3 kinases, a sodium proton exchanger required at the next stage. Activation of RAC occurs, PAC is activated, and then another set of kinases and other factors are involved. Eventually, then actin and actin associating proteins get activated, myosin 2 is involved, and then that leads to blebbing, and blebbing leads to endocytosis, and that in fact in turn leads to infection by Vaccinia virus. Now, what type of endocytosis is this? Is there anything similar known? One clue is that when you add the virus to cells, it starts to gulp in fluid, just liquid from the outside. It is the yellow line here. Normally these cells are all the time internalizing some liquid, but when you add the virus or a macropinocytosis activator, PMA, you get this huge increase in uptake of fluid. So that argues strongly, or starts to argue, that what we are looking at here is macropinocytosis, an endocytic mechanism that has been described already some time ago and is now more and more coming into focus. So what is macropinocytosis? It's a signal-induced transient endocytic pathway in most cells. It only occurs for half an hour or so. It involves ruffling and blebbing. In our case, it's blebbing. Other cases is simply ruffling of the cell surface. Those lamellopodia we saw in previous lectures are part of this ruffling event. The ruffles then and blebs, when they come back to the membrane, encase some fluid space vesicles, so it's an increased fluid phase uptake, all of it absolutely actin dependent. And it's characterized by dependence on RAC1, CDC42, and PAC1, and so on. Many of these, or all of these, were also found in our studies. So there is already a sort of diagnostic set of features which we know, and all of them fit with the uptake of Vaccinia virus. So just to go back to this one here, you see here is the macropinocytosis uptake process right here, and used by Vaccinia. We also know from other people's work it's used for adenovirus B, and perhaps in some cases even for herpes simplex virus. Okay. What is there in the virus particle that triggers all this? How does it actually manage to fool the cell into endocytosis by macropinocytic uptake? Turns out that the plasma membrane of Vaccinia virus is extremely rich in a phospholipid called phosphatidylserine. It's a lipid that is present in the plasma membrane of all cells, but it's on the inner leaflet. It's not exposed to the outside. It is known to be, however, exposed when cells undergo apoptosis. Exposed on the remnants of the cell, so-called apoptotic bodies, have exposed phosphatidylserine, and there it serves as the eat me signal, a signal that induces macropinocytotic and sometimes phagocytic uptake of those remnants by neighboring cells. So when a cell dies, the remnants of that cell are eaten up by neighboring cells, and that uptake process very often involves macropinocytosis. So here the virus also seems to contain phosphatidylserine, and if one blocks the phosphatidylserine with the phosphatidylserine binding protein Annexin 5, then the virus can no longer infect. And moreover, if you exchange the virus lipids, which you can do by first solubilizing away the other one, the original ones and then add new ones back on the virus, you can see that if you don't add any lipids, you lose infectivity completely. If you add lipids without phosphatidylserine present, you have also practically no infection. But if you put the phosphatidylserine back again, you can then practically rescue full infectivity again. So this and other studies show that the phosphatidylserine is critical for inducing the blebbing, for activating the pack, etc., and also for infection. So the conclusion from this, and I'm almost at the end, is that the virus here enters by apoptotic mimicry. It is mimicking an apoptotic body, and in that way triggering the response in this case tissue culture cells, which they normally would have to apoptotic bodies, which would involve all these complex events, including blebbing and endocytosis. So the virus mimics an apoptotic body. So if we go now through step by step what happens here, the MVs, these are the mature virus particles, bind to filopodia and surf along those filopodia to the cell body, probably using retrograde actin flow. The exposed phosphatidylserine then activates a signaling pathway which involves RAC1, PAC1 and other components. And the result of that activation is a blebbing occurs, first at the site of the virus attachment, but then globally around the whole cell. And then as the blebs are retracting, our view is that membrane is being internalized by macropinocytosis, and that brings in some of the viral particles. Once they are in the macropinosome, the vacuole that is formed, acidification takes place, and then the acid-activated fusion takes place in the macropinosome. So here is an example of a virus that does pretty much what I've been telling. It activates the cell. Without this activation, endocytosis would not occur, and it basically uses a complex cellular machinery intended for something else to enter cells. It's also interesting and important that macropinocytosis, unlike phagocytosis, is known to suppress native immune responses. That means that the immune response for the incoming virus is probably reduced, and that of course is in the interest of the virus. It's also maybe true that this virus, being so big, has had to evolve this different entry pathway because it cannot use clathrin-coated pits and so on. Okay, so I'd like to finish off by going through perhaps the most important implications of what I have been saying. The fact that viruses are Trojan horses means that they depend on the host cell for infection at every stage, from the early stages to the synthesis and also for final assembly of new particles. So what would happen if we would now start, instead of using antiviral agents and developing them against viral components, try to focus on making drugs which prevent the cell components from doing their job? We would then be able to block infection, like we did with many of these drugs shown here, and we would have a totally new situation, because viruses normally circumvent the action of antiviral drugs simply by mutating and becoming resistant to those drugs. It's clear here, I think, that it's very much more difficult for the virus to mutate in such a way that it can escape the use of critical cell components. Another good thing is that the cell provides many targets. Hundreds of different proteins are needed for any given virus. Any one of those, in principle, could be an antiviral target. We already know that if we look at the lists of these infectomes, some drugs already exist to some of them, and they may be developed further to have antiviral agents. Some of these targets are clearly present in the infectomes, or more than one virus, so maybe it's possible to imagine in the future drugs which block a whole group of viruses at the same time. And it will also be possible to have drugs, hopefully, which block each of the entry pathways, for example, so that when a newly emerging virus comes into the picture, a set of drugs maybe already exists, which blocks its entry pathway, because it will have to use one of the other of the known pathways. So I will stop there, and thank you very much.